Hello guys, today I would like to discuss the lab diagnosis of primary amoebic meningoencephalitis. So this is a disease that is often asked in microbiology university paper. The thing with microbiology is that you have to study the lab diagnosis of a very of lots of diseases. So this can be a little bit overwhelming and very hard to remember in your university papers. So this is some of the tricks that I used to do in my paper which helped me. So I hope it will help you guys too. So before I am going to start with the lab diagnosis part, I would like you to remember that this disease is caused by an free living amoeba, Naegleria fowleri. So this is kind of a little bit of fatal disease. 98% of people who have got this disease are dead. So this is a disease that will affect your brain. So when you start with the lab diagnosis part, you need a specimen, right? So this is a disease that is affecting your brain. So what will be obviously your specimen? It will be the CSF. So CSF is the specimen of choice, and you have to keep in mind that once you have taken CSF, you have to use it immediately. Refrigeration is not at all possible or recommended, as it is destroying the morphology of the parasite. So once you take out the CSF out, you have to look some things. So you are analyzing the CSF. So for CSF analysis, I want you to think the CSF as a soup. I want you to think CSF as a soup. So this is a protein soup, and the soup is very thick and purulent. Okay, thick purulent soup. So your soup is very thick and purulent. Purulent means pus cells, right? There is pus cells. Imagine you made your soup with lots of oils, and that oil represents the pus cells. So the pus cells present here is greater than 20,000 per microliter. Now this is a soup. You made this soup with chicken. What is chicken? Chicken is a protein. So protein, it is very high in protein. And then I want you to think that no one makes soup with sugar. So the sugar is less here. So when you are analyzing your CSF, the specimen you have taken out, it is very thick, purulent, and it is having increased protein content, decreased sugar content, and the pus cells here is greater than 20,000 per microliter. So that is your CSF analysis. When you are analyzing the CSF, I want you to think it of as a thick soup. Now I'm going to tell you the trick for your microscopy. For microscopy, I want you to think there is a bathtub filled with water in which so many amoebas are swimming. So this bathtub with water is representing your wet mount specimen, and the amoeba, it is the amoeboid form of the trophozoite. So when you take out the wet mount specimen, you will see the amoeboid trophozoite in it, and there is no cyst. See, and when you are using the microscope, it is better to use the phase contrast microscope. Next, I have a picture here where the torch is directly lighting into a amoeba. So the torch here represents the antibody, and the light here is representing the fluorescence. So trophozoid can also be demonstrated by direct. So this is directly lighting into the amoeba, right? So direct fluorescent antibody. By direct fluorescent antibody staining of the centrifuged CSF, and you are using here a monoclonal antibody. So since it is a monoclonal antibody, I have added only one antibody here. So two things when you are doing for the CSF microscopy, you have to first think there is a bathtub filled with so many amoeba. So wet mount specimen, you see trophozoid, amoeboid trophozoid, there is no cyst form, and trophozoid can also be demonstrated by direct antibody fluorescence or direct fluorescent antibody. Next is histopathology. So what is histopathology? You are taking a tissue and you are finding the pathology. Since this is a disease that is affecting your brain, obviously your tissue will be from the brain. So you are going to take a brain biopsy. brain biopsy tissue. So to remember that you are taking a brain biopsy, I have added a cute little brain here, and next to the brain you have a sky blue colored organism. So sky blue colored cytoplasm it is having, and it is also having a pink nucleus. So you when you take a histopathology, you need a brain biopsy. You're going to stain it, right? You're going to stain it with H&E or Giemsa stain. You will get a trophozoite. You will find a trophozoite who is having light sky blue colored cytoplasm, with a pink nucleus. So we have studied 4 tricks till now. I'm moving on to the next. So what is microbiology without a culture, right? So next one is culture. So the culture media here is you're using a non-nutrient agar. There is no specific nutrients in this agar, but keep in mind that for this amoeba to feed on, we are adding a bacteria. So we are adding here E. coli as a bacterial supplement. So this is my non-nutrient agar, and I have added a bacteria called E. coli in it. So what will happen? This amoeba will feed on this E. coli, and while it is feeding, it will move in the form of a trial. So there is a trial sign. I want you to use to remember this culture plate with a trial sign on it. And also I want you to remember this is a non-nutrient agar, and I have supplemented E. coli as a food for the amoeba to grow. Naegleria fowleri will eat and produce a trail. So next interesting thing for diagnosing or lab diagnosis is that this is my amoeboid form. Okay, so I am taking my amoeba and I'm putting it in a distilled water. Suddenly this amoeba is changing its form. It's transforming. Okay, this is transforming, and it has grown out so many flagellas. Okay, so since it is growing out flagellas when I kept it in the distilled water, this is called as enflagellation test. So you're taking some scraping from your non-nutrient agar, and you are transferring into a sterile tube. It is containing distilled water. As soon as you are doing this, the amoeboid form will undergo transformation, and it will form a pear-shaped flagellated form. Just remember, this is a pear shape. This is nowhere near a pear shape, but still, just keep in mind that in this picture there is a amoeboid form and a form which has grown out its flagella. Okay, so as soon as you keep it in the distilled water, it will grow out its flagella. So this was that. Then just like how we write for every lab diagnosis part, just write down molecular lab methods as PCR. And since this is something happening in your brain, you will do CT, MRI and all, right? So imaging methods like CT and MRI to show the obliteration of cyst. This is some inflammation, right? So cyst will be obliterated. So you can use imaging methods like CT and MRI. And before I have started this lecture, I have already told you that this is a fatal disease. Almost 98% of the people who got this disease are now dead. So no effective treatments are available, but using amphotericin B, or cute little bottle here, I have written amphotericin B on it. So our amphotericin B has some kind of anti-Naegleria effect, and 4 cases are treated. Till now only 4 cases were treated successfully using amphotericin B. So these are some of my tricks that I used to do, so I'll get my answer in my university paper. Just remember some of the tricks. Just remember that these pictures are there in my notebook. So when I'm writing my exam, when this question comes, oh yes, I have a soup picture here. What is soup? Soup is thick and purulent. So your CSF is thick and purulent. Soup is having protein in it, increased protein, decreased sugar, and I made it with lots of oil. So this is also having pus cells in it. Next, I'll remember there was a picture of some amoebas in the bathtub. So it is representing the wet mount specimen showing amoeboid form. Wet mount specimen will show only the amoeboid form. Then there was a picture of directly torch lighting into the amoeba. So trophozoite forms can also be demonstrated by using direct fluorescent antibody. Then next there was a brain and a blue colored trophozoite near it. So histopathology brain is to make you remember that you took the specimen from the brain and you stained it with H&E, and that time you got a blue colored trophozoite with a pink colored nucleus. Very cute organism, right? Next, I'll remember that I also had a picture containing a culture plate. So this culture plate shows a trial sign. So I have put a bacteria as a food for this amoeba to grow. While eating that amoeba, it produced while eating that bacteria, this amoeba produced a trial sign. Next one was our enflagellation test. I put my amoeba into distilled water, it produced flagella. And then finally you can add molecular methods as PCR imaging. Since this is something brain, if something happens to your brain, immediately doctor will order a CT or MRI, right? So imaging methods like CT and MRI can also be written, and this is a fatal disease, not much of a cure is present. So amphotericin B have some kind of nuclearia effect. You can write it down and you can finish your answer with it. So this was my trick for studying the lab diagnosis of PAM. I hope you find it helpful. If you find it helpful, please like, subscribe and comment. Also share this with your friends. Thank you.